What's up, everybody? Welcome to the EMS Lighthouse Project Podcast. MIke Verkest, Dr. Jeff Jarvis. It's going to take me 7 minutes to go through the supporting cast here.
I like it.
There's so much. But anyway, Dr. Emily Crow, hello. Thanks for joining us.
Hello,
and Dr.
thanks
Veer
for having
Vithalani.
me.
Howdy, how are y'all?
Oh, there it is. Okay, everyone take a deep breath. I said you. Did you not hear me?
When did you say me?
I said you like right off the bat.
I didn't hear you.
Oh, man. Everybody, please give a warm welcome to Dr. Goldilocks Ritu Sahni with his Michigan cutout behind him.
I have an audience today.
I was trying to figure out what that was.
It's my Michigan Stadium cutout.
Oh,
Yeah.
Since
Hey,
we
my
only
cousin
played,
Dan is on. I got to give him a quick shout out.
since we only played 6 games, and in the first time in the history of our stadium, we didn't win one at our stadium. They sent everybody their cutout.
I get the cutout now.
Yeah.
You have season tickets?
I do.
And since you couldn't be there, they put your little cutout up.
You had to buy it. It was like $40, and you send them a picture, and then they put it in the stands.
Got it.
Mike was supposed to do it for the Ducks, but he ended up not doing it. And I have like Portland Trailblazers season tickets, and so they're like, "Get your Trailblazer cutout." I figured
Now
one cutout.
Remley, what cutout do you have? Where's yours at?
My cutout's in a supply closet somewhere, but it would be wearing an Ohio State jersey, or should I say the Ohio State?
Oh, for God's sakes.
Anyway,
Oh, please.
nobody
Articles
likes
are
those
important. Come on.
guys. Nobody likes those.
I was going to get me a Texas A&M cutout, but sadly, I guess Notre Dame got in the way again.
I would have picked Texas A&M over Notre Dame.
I appreciate that. Notre Dame gets their butts handed to them, and yet they still get in there.
Yeah,
I think it's like religious discrimination.
it's
I mean,
something-
you to be fair, you got your butts handed to you by Alabama too.
Like the number one team in the nation that so far everybody who they have played.
Well, Clemson's beat everybody except the time they lost at Notre Dame without the best player in the country. I mean, Trevor Lawrence was fantastic yesterday.
Yeah, he was. That was good.
He was pretty fun to watch.
It is true. It is true.
Hey, listen, before we get too deep in the jackassery,
Yeah.
I really want to make sure people know that they're on the right podcast or the right live broadcast, right?
Including perhaps some of the participants on this one.
Yeah, I'm making sure, Veer knows that we're doing mRNA vaccine literature research. Was this the right one?
That's what I'm here for.
So,
Are we bringing a
Well,
little?
there'd be tacos.
yes, there should be tacos.
It's
Somebody owes me
BYOT,
tacos.
though.
Eric Bauer owes tacos to everybody.
I agree. This may have been my fault. I was trying to click over onto Facebook so I could follow the comments, and I managed to click onto last night's. These are kind of running together, but the second shift one, I'm like, wait a second. It still says second shift, Mikey. I think you like Ritu more than me. You're playing favorites.
Well, it's clear he likes me more than.
And then I'm like, wait a second.
He thought we had the
even
second
has
shift
the
little
comments
banner.
from last week.
He thought we had the
Almost
second shift little banner. No, so to be clear, we had about 150 people watching that one. We won't get that many for this, I'm sure.
That's sad. That's sad.
Yeah. We had I think we're over 1,200 download or views on the YouTube.
Yeah,
That was
yeah,
great.
good one.
I think that was a good, and it was good because you know why? Because we brought somebody in who knew his stuff.
Yeah. Yeah. No.
He was a very personable young man. So
Well,
Yeah.
we're doing that here too. We brought in Drs. Vithalani and Crow.
Yeah, we
As
got a
opposed
couple
to
of
the three of
big
us.
time pinch hitters coming in here. Pinch hitters makes it sound derogatory. Like we brought in the closers here.
we brought in the pros from Dover.
That's right.
For those of you who are our age or older. No.
We did have right before we went live, we're in the green room and somebody was mentioning the year they graduated from high school. Might have been the year I graduated from medical school.
And let's ask the audience, who do you think is the youngest on the panel here? Throw it in the comments. Let's hear it. I want to hear what you guys think. Let's see what we've got.
Who's the oldest?
Well, we don't want to do that. That's just rude.
Oh, yes, we do.
Oh, so Eric says nice MASH reference.
That's the pros from Dover.
I know exactly.
Yeah.
Pros from Dover. Yeah, a couple. No one wants to say who's the youngest. They're all a bunch of babies. Oh, Vithalani, Will Mercer.
Vithalani, we got to vote for Vithalani.
Yep.
Mercer works in my office. He knows.
He knows that Veer is 19 years old this month.
I see Dave Lincoln's there. Lincoln sucking up. Good
He's
job.
still my friend. It's okay.
Good job. Dan Oder might be right there. Could be right. All right, guys. So
somebody says the dude on the left. Is that me?
Yeah, no.
That would be. It's our left. It's my left.
I'm looking at the Facebook thing. I'm on the left there too.
Where?
Unless they're talking about me.
Yeah.
What does that mean? Did I just use my thing? Did I use my one? We talked about the cursing rules.
No, you're okay.
All right.
Well, I do think we need to cover the obligatory what's in your cup.
Yeah, that's true. And we got to hurry because you know what? I want to keep these people watching. We have a giant agenda and we have picked a lofty topic, right?
We even have an agenda.
Like, yeah, we actually had an agenda.
I know it's not an issue of.
Oh, come on.
I know it's not a second shift episode because there's an agenda.
Yeah, it's definitely not a second shift episode. No, we've got a lot of stuff to talk about, but we always have to do what's in the cup. So I'll start since I'm the guy running the joint. I made a Manhattan, but I cheated a little bit. I used Maker's 101 for that, and I made it a double because I don't have time to go make another one because we're busy. So that's what I got. Jeff?
Glenlivet 12 Sherry Oak.
All right. Ritu?
I too made a Manhattan. I was inspired by your Manhattan.
All right.
I actually made a triple, which was a little like it was this much. I'm like, honey, do you want a Manhattan? And she's
Baby,
like, yes,
I should have just done it.
So, and then, but it's with a Westward, which is a local distillery, but it's a Westward whiskey.
Maybe they're going to be maybe they'll be the new sponsor for the Second Shift podcast.
You know, I would love it if the old Westward hopped on with the McMenamin boys.
Yeah, we're not going to talk about
McMenamin brothers to be our sponsors because, you know, we haven't been at the place in over a year.
Yeah, it sucks. And Bremley, I'm noticing your fancy microphone that you got there.
I do have a fancy new microphone.
Is that the new Shure podcast one that they came out with recently?
Yeah, it's the new Shure USB one.
Woo wee!
Woo! Had to
That's
up my
good.
game to join you all.
I know, right? That's really good. Well, are you drinking anything, or you just got what do you got?
Maybe it's coffee, maybe it's wine.
Okay, copy that. Perfect. Well, if it was wine, what would it be? Something from the Willamette Valley, maybe?
Maybe a Malbec.
Oh, maybe a Malbec. Okay. Those are good. All right. Veer, what about you? Actually, we know, but we got to ask.
I'm going with my daily drinker, which is a Coke Zero over ice.
Oh, copy that on ice.
No lime.
I do have the Texas version of that, the Diet Dr. Pepper over here.
Yeah, I've got a backup.
I have the Texas version of that, which is the Texas Bubbly.
Okay. So good.
Very good. Very good.
All right. Let's listen. We got so much stuff to get into. It's been fun. It's been 10 minutes. Time's over. Time to get down to business. You guys agree?
You bet. Crack the whip, Mikey.
All right.
I hear you got some pictures for us.
Yes, I do. And so one of the things that we talk about, obviously we're going to be talking about literature, the research. What does it say about these two vaccines? But one of the things that we wanted to do just real quick, and I've got to set it up while we're sitting here, and this is going to just take me forever to do. But I wanted to get in and we wanted to do a little tribute to the folks that have gone out and got their vaccine this week. So I'm going to send this live and let's watch this quick video. It should only take us about a minute. All right. A little something there for the people.
I like it.
Yeah.
I like it.
And those were all folks that either Dr. Jarvis, Dr. Sahni, the crew reached out and said, hey, you got a picture of yourself? Send it in. We're just going to do a quick little thing. So that was not everybody because the picture started rolling in all at the last minute. I didn't have time to update it, but pretty damn cool.
Absolutely. We'll get an updated version out to our friend Hillary Gates with EMS World, who wants to run that too. Strong work, Hillary. Yeah, tons and tons of people. So I think we probably ought to check in because I know everybody's worried about what's going to happen right after you get that injection. I asked Christy, we turned the lights off last night. I was not glowing in the dark. No arms from my forehead. As a matter of fact, I feel fine. I know a lot of people, or at least some people, are feeling some symptoms. I barely even have any soreness. I think I did great with it. Veer, how about you?
Yeah, it was interesting. I sort of forgot that since it's such a small quantity, they're using our site was using an insulin needle, so I actually barely felt it going in, and I was glad I had the video showing it going in. Otherwise, I was sort of doubting myself for a second. First couple hours felt a whole lot of nothing, and then developed a little bit of arm soreness. Little mild headache that went away on its own after a few minutes, and then next day, little arm soreness really only when sort of lifting it, and by the end of the day, only if pushing on it. And today I'm fine. I got mine on Friday, so I'm 2 days in, and I'm pretty lucky.
Ritu?
Well, I swear the guy doing mine used a 14 gauge 4-inch needle.
That's because he knows you.
I guess he picked it up. I'm like, that looks a lot thicker than the needle I usually get my flu shot in. So, but other than that, my arm was fairly sore yesterday. So I'm 2 days. I had mine 2 days ago.
Yeah.
I kind of planned on. I got lucky timing wise. Was off yesterday, so just planned on an easy down day and relatively sore yesterday. But today, nothing.
Yeah.
But I do certainly know a couple people who are feeling kind of crummy today. The 2 days afterwards. So it definitely still happens.
Yeah, our buddy Dr. Troutman from Texas Tech was feeling kind of puny. I haven't talked to him today. I hope he's feeling better.
Yeah, and it makes sense. What makes you feel crummy is an immune response. So there you go.
As a matter of fact, I was texting Ritu. I'm like, man, I'm worried. I must have gotten the placebo in this thing. I don't feel anything.
Yeah, no, I agree. I was like, meh.
Absolutely. Well, you wanna hop on in? There are the two things we want to cover today are the two clinical trials. Obviously, the two EA approved vaccines, the Pfizer and the Moderna. But I'd like to do just a real quick summary for any of y'all who missed Dr. Smith's great description of what mRNA vaccines are. I definitely urge you to go back and watch that. He did a great job talking about what those things are. But I'd like to just give us an idea one more time about, one, how incredibly amazing the scientific breakthrough was here. Actually, it was probably more of a logistics breakthrough. The science breakthrough came a while back when we figured out what to do with mRNA vaccines. But we first sequenced the genome of this virus January 10th of this year, of 2020. It's still 2020, and we had needles in arms. That is absolutely amazing. 11 months from first sequence to highly, highly effective vaccines. And, when we talk about how quick it is, we're going to talk about this a little bit more. It's quick for a couple of reasons. The biggest of which is the technology, the ability to use these mRNA vaccines. And then the next thing is throwing just blank checks at this problem helped a lot.
Yeah, that was one thing that we talked a little bit about. And I can't remember. I think it was Dr. Sahni and I. It was offline, but I'm like, because the discussion with Dr. Smith sort of raised a lot of questions in my mind. Being, I'm just an EMS goob, right? I'm learning a bunch of stuff, and I'm like, what was so unique about like how this one went down? And because the process was the same, but the difference was the handwriting on the checks. It was money
Yeah,
was
and
no
I think-
object, right?
And the number of zeros.
Yeah,
Yeah,
the
and
number
I
of
think-
zeros.
Yeah, and I think when we talk about sort of other vaccine trials,
Mhm.
we'll be able to sort of kind of
Yeah.
parse out why there was the difference.
Perfect.
Well, let's talk just quickly about what fundamentally is different about this. And I before we do that, I do think we need to say that corners were not cut on this. When you went through these trials, you still did the same phase 1, phase 2, phase 3. They just ran them at much bigger scale. But let's talk about the technology, because I think if you understand that, you can understand one of the reasons we're able to do this so much faster. So this is an mRNA vaccine. And to give you an idea, all of the genetic material that codes who we are, that lives in the nucleus of our cell in DNA. DNA, not RNA. It's a different thing. RNA is how we go from DNA to actually building stuff like proteins. And that's really what we do. We crank out the machinery of life is the ATP pump and creating proteins. So the protein that we want to do, normally what we would do is make a virus in a lab and just generate a bunch of target cells, like say spike proteins. And the logistics of that take a really long time. In this case, we just decided we're going to let the body be the 3D printer, the biologic printer, and we supplied the instruction set, and that's mRNA. So we wrapped it in a little teardroplet. I heard somebody today say it's a little teardrop of butter.
It's like me.
Lipids.
It's like me. I'm a little teardrop of butter.
You're a big teardrop of butter.
Oh,
Mike,
thanks. That makes me
don't sell
feel
yourself short.
You should have let me.
What I
say-
said, that Mikey V, he's like butter.
So just a little bit of fat so that you can get it into the cell, and also so that the mRNA is sort of, I don't really want to say designed, but its job in life is to make proteins and then dissolve. So it has a very short half-life, and outside of the cell, it just dissolves. So we got this little bitty lipid piece of butter that slides it into the cell, and then the cell reads it and just starts cranking out protein. The protein is the spike particle, the spike protein, and that's sort of its little antenna. That's how people know what it is. And our immune system attacks that spike protein and creates antibodies, and it creates T cells, and then these little memory B cells. That's it. That's how it works. So I think about it as a biologic 3D printer. And it's wicked cool because instead of having to go through all the logistics of creating these proteins, we just have the body do it. And our bodies are incredibly effective at doing it. Millions of years of evolution work. So we just co-opted it and said, hey, why don't you spit out some spike proteins? So that's how the vaccines work.
That's it. Thanks for joining, everybody. Have a great night.
Yeah.
Drive safely. Don't forget to tip your wait
Tip
staff
your
on
waiter.
the way out.
Try the veal.
Yeah. That's,
Never try the veal.
I mean, I guess at the end of the day, that's really it, right? And I think one of the pieces that, Dr. Smith so eloquently put, well, I guess for me, again, I'm sort of the simple-minded guy of this group here. All these smart people, that one. Because when it came time to talk about some of these other long-term effects and these other things that people had to say, was that if you understand the mRNA part, then it would lead you to believe that for the most part, I mean, it's gone within a few days. There's no so therefore there wouldn't necessarily be long-term effects from that piece of the virus that am I sort of close and hovering around that?
I'd say you pretty well nailed it. Yep.
It's like a Snapchat.
Yeah. Isn't that great? Yep.
Yeah. That's a good way to put it.
And then it's gone. Yeah.
So-
No, it's it's nice and simple. Now, saying it's nice and simple, the ability to come in and create that little bit of mRNA that codes specifically for that spike protein and nothing else. And this is what the real breakthrough for COVID is we were able to stabilize it with this little butter drop, and it was stable enough to get into the cell. And as soon as it gets into the cell, we're home free. The mRNA itself, again, not DNA, and the mRNA doesn't go anywhere near the nucleus. It doesn't have access to the DNA. So it's not. Yeah, go ahead, Ritu.
Oh no, I apologize for jumping, but I mean, and that makes total sense. But there's lots of times where we thought something would turn out some way in medicine and it didn't, and so I think the next important piece is the discussion of these trials, and how we went beyond just saying the science makes sense. You know, let's put it into practice.
Absolutely. My favorite analogy on this is the science says that butterflies can't fly or bumblebees, yet they do. So sometimes you just have to look at the clinical evidence. So that's a good point. That's kind of what we're here for. Shall we talk about some clinical evidence?
I mean, if you want to, if we can get Dr. Vithalani to stay on for more than 12 seconds at a time.
I know this is why I was tap dancing over here. I'm like, Veer's up next, but Veer seems to have some connection issues.
Yeah. I apologize.
I was rereading the Pfizer trial.
Yeah, just in case. Yeah, so it's always
last minute.
Yeah. Well, let's quickly get through this before it happens again, shall we? So. Yeah, the Pfizer trial that came out, I think the initial data that everyone looked at first was their massive data dump, right? That comes as part of their EUA. So they put out their dump of data, and then the FDA comes in and writes their own brief that takes it and reshuffles it and then puts out the FDA brief. And I think everyone sort of preferred to read the FDA brief because it had commentary from FDA folks on it, which I think is appropriate. But then Pfizer and BioNTech put together an article for the New England Journal of Medicine that was published. And I think that's mostly what we'll cover because it's in a little better of a digestible format. I'm going to try and give, the right level of detail. If anyone wants more, let me know. So what Pfizer and BioNTech are doing, they're actually doing, it's one trial technically, and it's a phase 1, 2, 3 trial, right? And so the phase 1 data where they inject normal, healthy people with the vaccine.
Is that it? Is that all we get?
Well, I think we have uncovered a side effect that we didn't recognize of the vaccine.
Poor guy. This is like the fourth time.
Does he have Jamie Kennel's?
Yeah, he's got Jamie Kennel's
for those of you who watched
Kingston County Fire District 17 Wi-Fi signal, unfortunately.
Well, he's from Fort Worth, so we didn't really introduce him, but because we all know him so well. And as way of stalling completely transparently here.
It looks like he might be back in the green room here. Let me check.
He is the medical director for MedStar, which is the ambulance service, the ambulance authority for Fort Worth and the surrounding areas. But Fort Worth is a great town, but the downside is they do everything by horseback. So the horses that are running the little electrons back and forth to his internet signal seem to be a little distracted right now.
Well, and the thing is it's not his signal, it's his computer, as if you might recall. He was having an issue with his computer.
All right.
Restarting, so someone's going to have to take over.
All right. Well, let's
and it ain't going to be Mikey V, I will tell you that. It ain't.
Let's talk a little bit about it. I got some notes in here somewhere. I'm going to have to put some reading glasses on. So I'm just going to go over the New England Journal of Medicine paper because Veer was a little bit subtle about it. The FDA dump was indeed a dump. Tons and tons of papers, but they published this. Hopefully you can't see that at all. It's a New England Journal of Medicine. The title of it is Safety and Efficacy of the BNT 16.2B2 mRNA COVID-19 vaccine. So this is a paper from Pfizer that summarizes their findings and the mechanism. So if we'll go over it real quick, I will do basically the PICO question here. So the population intervention control and outcome. Mm-hmm. So this is a study it's a multinational randomized controlled double-blinded trial. And they had the largest country was the US. We had the most sites. I want to say then it was Argentina, and then maybe Brazil. There was also Turkey and South Africa. I may be forgetting one, but those were the big countries that were involved. And the population were patients in those areas who did not have COVID before they were enrolled, but who were at risk for having COVID by nature of being alive on this planet right now. They were 16 years of age or older, and the only real exclusion was pregnancy. So that was our population. The intervention was their mRNA vaccine. And the mRNA vaccine here is, again, just a little piece of sequenced messenger RNA coated with this little lipid nanoparticle coat, which is a big fancy way of saying butter. There are no, people always talk about, all the additives and adjuvant medications that are added in to make the vaccine work better. There are none of those in either one of these. It is just mRNA and the surrounding area. This ends up with, the dose of this ends up as 30 micrograms. It's about a 0.5 ml of solution, and it's given twice, once on day 1 and then again on day 22, so 21 days apart. The big challenge with this vaccine is that it has to live in the Pfizer freezer. I'm trademarking that, as y'all know. Pfizer freezer is my term. It has to be really, really cold to maintain the stability of it. So that's the intervention, 2 of these doses, 21 days apart, and then the control was a placebo.
Look who's back.
Veer.
I am so sorry. Now I've officially switched to a new computer.
There you go.
I hope this one stays on. I bet my desktop will stay on now from this point forward.
Of course.
But I apologize. How far have you gotten through?
So we were, I just switched over and I was going to talk about sort of a PICO question, population intervention, control, and outcome. We have talked about the methods, the population, the countries involved, what the vaccine is, the intervention, the control, and I think we're ready to head over to what their outcomes were, meaning what they were measuring, what they were looking for, and what they found.
Okay, great. So, I think it's important to understand this study is technically, really, it's broken up into 2 parts. There's the safety component and then the efficacy component, and you'll notice that throughout the paper. So the safety endpoints, they really had a couple primary endpoints. The first was within 7 days, and now they took a subset of patients and they asked them to answer questions every day based on how they're feeling for up to 7 days after each injection. And that was the first safety endpoint. And they were looking for solicited, meaning they're going to ask you what symptoms you had and really going based on the ones that they found in their phase 1 trial for both local and systemic effects. So things like fever, headache, fatigue, allergic reaction, et cetera. And then whether or not any medication was used to manage those symptoms. And then for the overall population, not just that one subset, they were looking for up to a month from the second dose any unsolicited adverse effects. So any big effects that happened that they didn't ask about, but they would expect that they would get reported. And then for up to 6 months after the second dose, any serious adverse effects. But because of the data cutoff, they only had 14 weeks of data for the majority of people. So they had 2 months of data for everyone, and they had 14 weeks at the most after the second dose for this one other sort of subgroup. And then they put in this thing called a stopping rule because a lot of people have been concerned, well, if you've got previous infection or if we vaccinate you and you develop antibodies, and would this make your disease, if you were to get it, worse? Would it be more likely to give you a severe outcome? So they put in this thing called a stopping rule that they mentioned, and it basically says that if you're 5% more or equally as likely to get severe illness, then they're going to stop the trial, and that at 11%, they're going to watch for it. So that was mainly the safety aspects of the study. From an efficacy perspective, the primary endpoints were, was it effective against confirmed COVID-19? And then they go on to describe what confirmed COVID-19 is from 7 days after your second dose of injection. That's where their cutoff was. For people that didn't have previous infection, and that's an important point because there were some people that ended up getting vaccinated or in the placebo group that did have previous infection. But that's their main primary endpoint.
And then they had a secondary primary endpoint, which was everyone, which is nice because it turns it into a pragmatic trial. It says, well, if we just vaccinated everyone, what are the outcomes going to be there? So whether you had previous infection and your own antibodies, were you likely to get reinfected or not? So that was their primary efficacy endpoints. And within that, they defined COVID-19. Yeah.
So you said their primary outcome was whether it protected against COVID or not, but can you define what they meant by COVID? Is this an asymptomatic positive or?
Yeah, great point. So COVID-19 was actually, the definition was actually determined by the FDA, not by Pfizer or BioNTech, which is, I think, a really useful point here. And basically they said you have to have a positive test, that's the first part, and you have to have some symptom, at least one. So they go on to say fever, new or increasing cough, new or increasing shortness of breath, chills, sore throat, diarrhea, vomiting. It's all listed in there, but you have to have a symptom plus a positive result, okay? And then they talk about the same FDA criteria for severe disease is meeting the definition for COVID-19 plus some sort of reason that they would tag you as severe. So either you're in respiratory failure, evidence of shock, admission to the ICU, that sort of thing. And that was actually their secondary endpoint. Was they're not powering it to look for that, but they're also going to evaluate, was there an effect on severity of illness if you did get it? So those were the main sort of outcome measures that they were going to look for, both for the safety part of the study and the efficacy part of the study.
And I think their definitions on that were, most of us would look at them and go, yeah, if you're in the ICU, if you need not just oxygen, but high flow oxygen, I'll spot you severe on that. If you're in shock, yeah. If I have to tube you or put you on ECMO, I mean, ECMO's kind of borderline. That's kind of went.
Yeah. Well, and it's interesting, if you do wade through the actual EUA or the FDA brief, it goes into a little more detail and it talks about this SpO2 cutoff of 94%. So if you had an SpO2 of less than 94%, you were deemed severe. And the one case, skipping the results, the one case of severe illness in the vaccine arm had an SpO2 of 93%. So, technically it was severe, but I think by current all standards, it's not severe. You'd still go home from the hospital with that. So,
I think maybe they were confusing respiratory distress and pre-oxygenation before intubation. They just wanted to give you a little buffer on there.
Yeah, exactly. If only the FDA understood all that.
Yeah.
Yeah.
So Veer, how big were the groups?
Yeah, so they overall, I love result trees because they do a nice job of elaborating everything else. So between July 27th and November 14th, they screened about 44,000 people. They had 43,500 or so, which ended up getting randomized. A couple people had left for other reasons. And so each group had, 15,000-ish across both of them. So some in the placebo arm, some in the vaccine arm. And then it continues on sort of down the tree. Some people ended up getting sort of removed from the trial before vaccination, so they had a reason to not continue. Then they separated those into the randomized group to get the first shot, whether it was vaccine or placebo. A few more sort of flittered away after that. Then those got the second shot, and then they followed most of those people for a minimum of 2 months for them to be able to judge that efficacy data. And again, that 2 months was decided by the FDA. Because they wanted to that was their sort of threshold of data that they wanted to see. So in total, about 18,500 got the second dose, I think is the sort of final ends that we're looking at in each side. And the nice thing is they did a decent job with their demographics. So it was pretty much half female. There was about 57% or so almost half in what they called the younger population, which was 16 to 55. And then 42 or so, which were 42%, which was in the over 55 group. Everyone likes to call out the fact that there's 35% of patients which were obese, which is actually probably a representative sample for most of the country. And then they had about 20% which had pre-existing conditions, which I know we've talked about pre-existing conditions a lot, and there's probably more 60% in the overall general population. There is a little skew there. And then there was a pretty heavy predominance of white participants. It was like 80-something%, which I'm sure when you compare to the Moderna trial, there was a little more uptake in the Moderna trial across the sort of racial spectrum. That's pretty much what we're looking at.
So that issue about, first off, I would like to point out that the two oldsters here, me and Ritu, we clearly.
Okay, I mix up you white guys all the time too.
We're still, we do all look alike. both of us, we are still in the, I'm trying to compliment you, dude. Help me out here. We're both still in the young group. In both trials, we're young. We're still in the young group in the trial. That's correct.
Absolutely. Not by much.
Well, some more than others, but yeah.
You know what though? I got a question when you get done with your thought. I just got to get it in.
Yeah, so I was going to talk a little bit about the challenges of recruiting what they call here communities of color, people of color, minorities.
Yeah.
It's a challenge because we recognize the challenges of running trials only on white men between the ages of 24 and 26, because that is not necessarily representative of women, of other ethnicities. So it's a challenge. We would like to be able to say, well, we want a population that we study that is the same as the population, in this case, of the planet. Well, we're dependent upon volunteers. So you have to have people who are willing to volunteer, and different communities are more willing to volunteer than others. And this is different in different places in the world. In the United States, African Americans are much less likely to volunteer. And there are probably some pretty good reasons for that historically. I mean, Tuskegee
some real good reasons
come to mind. Some syphilis trials. So as a function of the different populations involved, it's a challenge to get to it. They did a decent job here, but it's still not as good as we'd like. But it's what they could get.
I'm curious if Dr. Crow has any thoughts on that.
Well, I think Jeff hit them that, yes, ideally we want to recruit as generalizable as possible. And sometimes there are some challenges, and there's good reason why IRBs exist, and some of that distrust is still present today.
It's good reason why you don't have a whole lot of, fortunately, there's no reason to believe physiology is different between prisoners and non-prisoners, but good luck getting IRB approval to do studies of prisoners.
Yeah.
So you're going to have to just, and for good reason, I might add.
Yeah, it's all question. I mean, one of the key components of doing research for volunteers is you have to be doing this of your own free will, and you can't.
Are you saying prisoners are not of their own? No, they're not. Thank you.
Yeah.
Somebody comes to you and says, "You're a prisoner. Would you like to be in this trial?"
Yeah.
I don't know that that's a free will decision.
Would you like to be in this trial?
I grew up just outside of Huntsville, Texas, which is sort of the head of the Texas prison system. And boy, were we into the prison rodeo, and would say, well, they're just volunteering.
Okay. I got to get this in. Because Nick Adams is killing me. Have they researched the effects on males with super hairy upper arms? Listen, jerk. I am one of those people.
Well, I think he might be referring to the two folks that got their injection through their sweaters.
My two other brothers here. Listen, sometimes it's just the way it is. Okay.
I mean, it is. I mean, if you're deficient in testosterone, you would put that comment down, Nick.
Right.
The injectable kind doesn't count.
Jeez.
I'm sorry, Veer. You were going to tell us about what we actually found. Sorry for the distraction.
No problem.
Second shift on you there. Sorry.
Yeah, we went a little second shift. That's good.
All right. So when we get into the results, so there's again going into that safety and efficacy arm. Within the safety arm, there was that they call it the reactogenicity subset. And best I can understand, because they were kind of vague about it, it was 8,000 or so participants that got a survey for 7 days after their injection, whatever it was. And it was not the entire group. And they were asked specifically, do you have these symptoms today? So it was the ones we talked about: fatigue, headache, systemic effects, etc. And within those 8,000 ones, they looked at local, meaning pain, redness, swelling, that sort of stuff, and then systemic. And basically, there is an increase in local reactogenicity, and the majority of that is mild to moderate pain at the injection site. And less than 1% of people had what they called severe pain or described that their symptoms were severe. It was less frequent in that over 55 population, and the majority of them resolved within 1 to 2 days, which is consistent with, it sounds like, the experience of those of us on the call who did go through this. And then within the systemic reactogenicity set, so again, ask the question, hey, did you have these allergic reaction, lymphadenopathy, fatigue, headache, that sort of stuff? Again, it was more likely in the vaccine subgroup, more likely in the younger population, and the majority of symptoms were fatigue and headache. So again, within those percents, it was 59% for fatigue and 52% for headache. So you are expecting that, and again, it's mostly after the second dose. But the rate of severe events was less than 1%. And so in general, that was pretty rare. When you get into post-second dose, it was less than 2% of people had those sort of severe symptoms. And then they looked across the entire population, so all 43,000 participants, for adverse events. And so this was unsolicited, that it was found out or it was reported to the study coordinators. And so that was the lymphadenopathy, any serious adverse events. So you'll see things like somebody got, it sounds like, injected into their GH joint, I guess, or had a damage from that. Somebody developed like a leg limp, I think, is what I saw. And then a couple people died, 2 in the vaccine trial or vaccine arm and 4 in the placebo arm. Pretty much everyone said the deaths were not, or the investigators and the Data Safety Monitoring Board basically said the deaths were not attributed to COVID-19. In fact, none of the study participants died from COVID-19, which is also kind of interesting when you look at the overall death percentages for COVID. But on the whole, there is some increase in potential systemic effects, but you have to compare it to the rate of things in the general population. And I think that's where things don't really get pulled out a lot in the paper itself, but it's mentioned in the EUA documentation of, well, hey, this happened, but compared to the general incidence in the community, it was actually lower.
Yeah.
That pretty much sums up the safety part.
Yeah, I think that's a great point. And it is something that people will tend to forget about, is that people still have their normal everyday, whatever it is that's going to happen to them, whether they're in a trial or not. And
there's one in particular that I think Jeff's going to talk about with the Moderna trial that is getting a little bit of press.
Yeah.
Interestingly, nobody in this trial had a allergic type reaction of the, was it 18 or 19,000 that ended up getting the vaccine, which has now been reported since then in I think 3 or 4 cases now.
So there are a couple. Yeah, right. So the question is this vaccine more likely to cause anaphylaxis, which is what we're really concerned about. If you get hives, is that an allergic reaction? Yeah, it probably is. I'll take the benefit of the vaccination over the hives that will get better with Benadryl. Anaphylaxis, well, you got my attention there. But it does not look like, and I didn't look at the numbers that closely on this trial. It wasn't there like at all in either group in the Moderna trial. So we're just not seeing it. So there are 2 reasons for that. We had 40,000 people in the Pfizer, is that right? 40,000 total? And
yeah, 40-ish.
30,000 in the Moderna. So 70,000 people now have gone through it with 0 reported anaphylaxis. But we're talking about immunizing around 200 million people in the population. Just raw numbers says you're going to have somebody with anaphylaxis. Whether it is attributable to the vaccine or not, you're going to have someone temporally related. So I think it makes sense at the vaccine delivery places to, also be able to be an epinephrine delivery place. We know that anaphylaxis is just Latin for I need more epinephrine. There's a fix for that. We need to make sure we have it, not to make light of the consequence. But I think the rate that we're seeing it, like not detectable in the trials, is pretty limited. The other issue is if you had something in the trial, they're going to report it, but it is a controlled trial and they are really going to do a lot of investigation to report it correctly. The CBS News ain't going to do that. The New York Post is going to see, well, you're going to see something. Somebody had anaphylaxis, and what they really had is a vagal syncope. So the definitions get a little bit, it does get more probable that it's going to happen just because more numbers, but then your definitions get really loosey-goosey when it hits the media.
Yeah. All right. Well, good. So you want to sort of round us out here on this, Veer? What was sort of your take home on the on the Pfizer?
Well, let's hit
So let me just
the
hit the efficacy results real quick.
Right,
Yeah. So, and I think that's the part that, people do care about both aspects of it, but I think, they're equally important. So they had about, remember the primary subset that they were really looking for were people without history of the disease. So in that overall group, there was about 36,500 or so that had no previous infection. In the vaccine arm, there were 8 cases. In the placebo arm, there was 162 cases. And so I think, through the fantastic math of the world of statistics, they got to that 95% vaccine efficacy. And I think we're going to touch on that a little later. When you look at people who either had or did not have a history of COVID, it went up a little bit, 9 in the vaccine arm and another 160 in the placebo arm. And when you break it down by really every subgroup, whether it's by age or by ethnicity or by comorbidities or everything else, the efficacy was basically the same. And then a couple things on top of that, it wasn't powered for a 1-dose evaluation, but it looked like there was about 52% effectiveness in that after that first sort of dose. And then that protection started about 12 days after that first injection. So it's not great, but there's something to be said there. And then it does look like there is some protection against severe disease, but again, not powered for that, and they weren't able to really look at that. The things that we talked about.
Hey, Veer. Yeah. So that 50, I don't know if something's.
Yeah, I'm glad it's you this time and not me.
That was weird.
So the 52% piece, I don't know if you guys want to talk about single dose effectiveness now, or you want to wait till the second trial is discussed. But when you look at the graph on the Pfizer trial, I think part of the reason why you're right, it's not powered. But if you look at the number of cases in the vaccine group, in the not the control, but the actual, there's not like an inflection point after the second dose. There were very few cases after the first dose. And the reason why it's only 50% effective is because it was only 2 weeks out or 3 weeks out from the initial. So there wasn't, and I know Remley, hopefully we'll talk about this because we talked about this in the chat, but the idea of like, how do you measure effectiveness? And I think part of the reason it was only 50% effective was because of the definition of effectiveness, and you hadn't had enough time yet to build another more positives in the placebo group. I'm not arguing that we should only do 1 dose, by the way. I'm just saying that there might be some hint of information there that argues that you could study this further.
Yeah, and the thing I found interesting too is we talk about that 14-day incubation period, right? I mean, there was nothing leading up to your first injection, right? There was no need to mandatorily quarantine or everything else to try and limit your ability to catch an infection. So you could have got inoculated on the day of your infection or your injection, and then carried that through for 10 days or so, and then all of a sudden you're in that first population group. So I agree. So only other thing I'll sort of comment in really a quick summary, and this is something that's been talked about a lot already. The study obviously looked at symptomatic cases, right? And I think that's sort of the big take-home point here is we are really aiming to evaluate, does it prevent you from getting symptomatic illness and to a lesser extent severe illness? It would be great if it prevents all infection, right? Whether symptomatic, asymptomatic, et cetera. And then within that asymptomatic group, are you infected but not infectious? And it sounds like looking into their data, they do have some information on that, and they are planning on resulting it sometime here soon. It would be nice if they had just tested everyone, the day before their second dose, and then we would have had the data at the same time. But maybe they did that and didn't report it in this study. But it's definitely the big talking point that I've noted. And then also, there were a number of people that were excluded from the trial, which for safety reasons makes sense. But it's hard to, you're basically best guessing when it comes to extrapolating the information to those other populations. I think we understand how an mRNA vaccine should work, but again, who knows what effect it has on these other groups.
So I've got a question for you. So I know we're going to talk about EUA versus approval here in a minute, but so if you guys all got the Pfizer vaccine, So are you? Is there sort of an ongoing study, including you guys? Because
Well,
it's
not a
EUA
study.
or
Sort of, but it's not a controlled study. And what I mean by sort of is that there is we're paying close attention to the people who get it. And we're doing everything, and I mean the FDA and the CDC are doing everything they can to follow the folks who are getting it. So I don't know about Veer and Ritu. When I got mine, I got
I got it. Yeah.
it right here, this nice little thing that probably y'all both got, and it's called the vSafe app. And app is a loose term. It's not an app, it's a website. And basically they said, scan this, so they know I got the Pfizer vaccine. They asked me when I got the vaccine, and then they will start pinging me every day. So I got mine at about 2:00 today, saying, how you feeling? How's your arm? Is it red? Do you glow in the dark? Do you need a doctor? Are you running a fever? And they asked some very specific questions, because what they're looking for is the prevalence, incidence of these the exact same adverse events that Veer was discussing and that I'll discuss with Moderna. So they're really going out of their way to try to track those things. But that's
standard in a vaccine trial, right? There are 4 phases in a vaccine trial, and the 4th phase is the ongoing monitoring component of all people who receive the vaccine. And so we are, and when we talk about EUA and approval, phase 3 is still ongoing. That's why it hasn't received final approval. And phase 4, though, which is the ongoing monitoring of both effectiveness, not efficacy, but effectiveness, and adverse event, is now in play, and we'll be there until we stop vaccinating, whenever that happens.
So let me address something. I was just looking in the notes here. I see Jen Casey's in. Hi, Jen. And she is asking about the difference between, so just looking at Pfizer, and all 3 of us got the Pfizer vaccine. Is there a difference between what we got, in my case, yesterday and what I'm going to get on January 7th? And the answer is no. It's 30 micrograms of the vaccine. The injection is absolutely identical. Well, her follow-up then is, why are the adverse events so much stronger the second time around? And it has a lot to do with what Veer was discussing about that damn it. See, there I go again. I can't tell you all apart. Ritu was talking about with the increase in efficacy even before the second dose. And basically your body, what we're trying to do is induce this immune response. We're trying to build up antibodies to the spike protein. We're trying to make T cells, and we're trying to make B cells that will then amplify up our ability to attack this virus. And that takes time. Well, between day 1 and day 22, we've hopefully done that, so that when we are again exposed to those spike proteins, we really attack it then, and we get more of an immune response. So that's kind of the reason for the differential response. I think there was another question. We can pretend this question is really about the Pfizer vaccine, although I don't think it is, and that's from Cody. Veer, you're going to have a hard time answering this question, same way I'm going to have a hard time answering it. But why don't you take a stab at it, because I do think there are 2 important parts here. Cody is asking about immunizing kids, because the thought is they may be more likely to be asymptomatic carriers and then transmitters. So I think he's going to be disappointed in our answer.
Yeah, I mean, I think the simple answer right now is you can't, because it's not under the EUA, right? So the EUA is only for 16 and up, and separating out the fact that we have no data, you're not allowed technically. I think if Pfizer can come out with some good data to show that it does prevent asymptomatic transmission, And, they did start collecting data from the 12 to 16 age group, which are the ones that look like they have, the highest rate of transmissibility within children. So I think once we have more data and once we know, what the safety profile looks like in kids, the problem is, and the pediatricians were saying that the difference between a 16-year-old and 17-year-old is negligible, but it's really pre-puberty and post, and we don't have any data for pre-pubertal children with the vaccine. And so I would love to reach a point where we can vaccinate our littles. I've got little kids and I think it's on the way to getting back to normal, but we definitely don't have the information yet. And we're not allowed.
Yep. Yeah. It's great question. And you answered it really well. Perfect. I want to come back to Samantha is asking about people who have already had COVID. And let's hold on to that because that's a really big question we can hit.
Some facts coming up. So, yeah.
Yep. Big issue.
And it looks like we have Freddie Mercury in the audience, too.
I am loving that. Yeah.
I wish people just use their name.
Who's Indian, by the way? Which I didn't even know till I saw that damn movie.
Zoroastrian.
I'm like, he's one of my people. One of our people.
Our people, yeah. So that's actually one of my EMS fellows, Dr. Farouk Makery, who many of you guys will know. The question about whether or not first responders and pre-hospital providers are characterized as healthcare workers, ACIP seemed to continue to move things around and decide where they're going to go. Texas specifically, for half of us, has included EMS and medical first responders within that top tier. The problem is there's a little bit of a functional differentiation inside that top tier between hospital-based healthcare workers and pre-hospital-based healthcare workers. I think now that Moderna is shipping this coming week, I think things are going to catch up. But right now, there's definitely been a functional preference towards hospital-based employees who probably some of them aren't in as direct contact with COVID cases every day as pre-hospital employees, but it has to do with the logistics, I would say, too. So at least in Texas, it's 1A.
Think about this as, and I do know some states have specifically said EMS is not in 1A. And I saw NAMSP and NAMT and ACEP all lost their minds about that, appropriately so, and put out position statements that said not so fast, buckwheat. So that's incredibly appropriate.
I just want
the other thing, the logistic thing though with Pfizer is, remember, you have to have these great big freezers. It's relatively easy for hospitals to do that. It's much more of a challenge for EMS agencies to do it.
And it's not just a freezer or a fridge. It is a data logger, and data loggers are hard to come by right now. They have to be certain temperature ranges. They got to have Wi-Fi and a proper certificate and that sort of stuff. So I agree.
Yep. I'm sorry, Mike, you were about to say something.
Yeah, thanks for running me over. That's fine. It's totally fine. Just a lowly paramedic on here. Remley, well, you're a doctor, so I guess it's different. But anyway, I'm the lowest guy on the totem pole. I was just going to say is that, in Oregon, EMS providers are 1A, pre-hospital providers are 1A. But I think we also, there's some assumptions there. I think, and there is some differences there. There's a delta between what 1A is and what 1A people think is, right? And I think we have to be patient, right? I mean, like there's only so much vaccine right now. We are definitely in the group. And this is a multi-week, couple month process here, right? And I know people are itching. I know they're chomping at the bit to get this. Just hang in there. We're going to get it. There's going to be enough for everybody. It's just, you're not going to get it 2 weeks ago. It's just kind of the way it is.
I'm personally really excited that there is this demand that people want it. I love it.
Yeah.
I got word yesterday that we should be getting the Moderna vaccine, and our medics should be getting needles in arms this week. Hopefully, like early this week.
I'm hoping that's something.
Yeah, we
like
heard the same.
What were you going to say, Ritu?
Oh, 1A is going to take 4 to 6 weeks to 2 months to get through. And so when something's going to take 2 months to get through, somebody gets it on the first day, and somebody's going to get it on the last day.
Yeah.
And the thing that drives that is logistics.
Right.
So, I mean, it's just going to, it's interesting because it feels like forever to people who want it, but
yeah,
we've been giving vaccine in Oregon since Thursday. So 4 days, or maybe since Wednesday, 5 days. Yeah, it's coming. It's definitely coming.
So I did notice that, and this is, again, it differs by state to state. Dr. Fagan there was just texting me and saying, hey, even as of today, 3:30 TDM call, they reiterated EMS was 1A. So I think it's a state by state thing.
Yeah.
Yep.
Yeah, there was one state that shall remain nameless that said that EMS was 1B because they didn't spend enough time with the patients.
Yeah, it was a time limit.
In a 4 foot by 6 foot box with no ventilation.
I think I just put out some data about that to show.
I think so. Dr. Crow, what did your data dump tell us?
Answering that question.
Yeah. So we did start to get some reports of this confusion with EMS and public safety. And there were about 10 states that had EMS outside of 1A very clearly in 1B or 1C. And so, as part of our work to try to advocate for the profession, we put together a little bit of data because one of the points was that, hospitals getting it because they spend 15 minutes with COVID patients. And I said, well, what's our average on scene to at hospital time? And then how much of that time is in the box? And so we put out the medians for that, both urban and rural, and we're talking 15, 20 minutes in the back of that box on average. And then some other good arguments came in. Well, what about PPE under less than ideal conditions when it's raining or they start to get wet? So I think that's some important data just to add to that conversation. I don't think it's the majority of states, but there still is a little bit of wiggle going on out there.
Absolutely.
All right. Well, we are at 1 hour and we are in literally agenda item number 2.
This is, I might add,
has nothing to do with my computer issues.
Well, I'll tell you what, let's, why don't we hop into the Moderna? I threw a couple of slides together, but I'm going to go over this pretty quick.
No, I can do it for us. Let me get us moved.
A couple of things that I'll say is MIke is clicking some buttons. There is, Veer pointed out that the FDA was driving these trials. So if you look at what I'm about to say and say, well, isn't that what he just said? The answer is yeah. The trials are very, very similar. There are a couple of differences, and I'll just focus on those differences. Methodology is essentially the same. This was an entirely US-based trial as opposed to the Pfizer one. And it again is 2 doses. It is 2 doses of 100 mics. And it is based at 28 days apart. So day 1 and day 29 instead of day 1 and day 22. Again, patients were excluded if they were pregnant. And here we had patients who were 18 or older was the inclusion. It was also an phase 3. And let's just kind of talk the grouping where Pfizer was 16 to 55 versus over 55. Here, they were 18 to 65, and they actually broke them up into 3 groups for stratification. And it was 18 to 65 healthy, 18 to 65 not healthy, and over 65. And interestingly, 82% of the people who participated in this trial were considered to be at risk for COVID because of an occupational exposure. 25% were healthcare providers. So these were folks that were more likely to get COVID. Ultimately, they're looking to enroll 30,351 patients, so 31,000 in this, so that's our N. And their outcome is identical to what Veer said. So primary outcome is the vaccine efficacy for preventing COVID disease. And they again define this as a positive PCR test plus symptoms. And for some reason, I always think about somewhere in medical school, I'm still trying to purge this from my brain, but minor and major criteria. You had to have 2 of the minors or 1 of the majors. to plus your PCR test to have COVID. And then severe is, again, what we would call severe. All right, so those were their case definitions. I would like to talk just a little bit. Actually, you know what? Why don't we have the person who actually knows about the maths? Rimley, you want to talk a little about the slide we have up there about what vaccine efficacy is?
Absolutely. Although I said that I felt a little guilty being invited to come on this one because I don't want to imply in any way that you need to have a huge background in statistics to understand the results of this, because these are well done RCTs. And so they did all of the hard work up front when they randomized the patients, and randomization works. So that lets us do really simple math. And now the easiest way for me to think about VE is just we take 1 minus the relative risk. And relative risk is pretty simple. It's the risk in the vaccine group over the risk in the placebo group. And so in the case of Moderna, what did we have? I think it was 11 over 185 was the final for the second analysis they did. And so 11 divided by 185 gives us between 5 and 6%. You take 100% minus that, and you get between 94 and 95%. And I think that they were putting out 94.5% efficacy. And so that's just a really simple way of doing it. And that math works because they're equal arms in the randomization.
You guys are so funny. It's so easy. You just do the math.
Two numbers.
And you're like, oh, what's for dinner tonight? Like,
the fascinating thing about
Tune back in. I'm done saying numbers, promise.
it's funny. We were talking about, the math is easy. And I don't know if I want to throw Rimley and myself under the bus, but we were trying to do some really simple, straightforward algebra earlier today.
Fractions is hard.
Oh my Lord, we were having problems with that. And ultimately we were trying to I was giving her crap about having an Android phone and needing the calculator on an iPhone. And then I promptly said, well, you know what? I sort of messed this up because I screwed it up too. And even with
who had the right answer?
Well, I'll tell you what. So I actually, good Freddie Mercury there, the good Zoroastrian, was asking about NNT. So NNT is, let me see if I can get this right, Rimley. It's 1 minus the difference. So instead of me doing math, I'm going to ask Rimley to calculate that real quick. Let's go.
But I actually started to go through this calculation the other day because I'm a nerd. But the NNT is actually huge because of the relatively low incidence of COVID-19 in both groups.
So
it's number needed to treat for those who are not done.
Sorry. Yes. You're right. This is not exactly one of those. I'm sorry. 1 divided by, not 1 minus. Thank you. It is, we typically do it in RCTs that are calculating the proportion of a group that has the disease. Well, if we look at this, the proportion that has the disease, there's a reason we're using incidence, which is the number per 1,000. So it's going to be a really, really small number. And
that's not unusual for vaccine trials.
You could probably still do it because both groups are really going to be small. Right. I'll tell you what, let's go over this and then I'll make a note and I'll try to do it just to prove I can. The demographics here, similar to the prior group, 53% male, median age is 52. I will say that in the 18 to 65 group, there is one very, very special person. My daughter, Sydney, was in the Moderna trial. 79% white. People of color, as they say, communities of color, were 37%. And if you're wondering why the math doesn't work quite right, you can be white and Hispanic. You can be white and non-Hispanic. For that matter, you can be black and Hispanic. So there's a little bit of overlap. That's the reason we calculate it that way. Go ahead and go to the next slide. We already talked about the occupational risk. And then 78% of folks here were healthy. The other 22% had a variety of conditions. And Mike, if you don't mind, move into the next slide if you can. Some of the conditions that they included, some people say, well, this is the best possible result and nobody will get this good in reality because they cherry picked who they gave it to. They really didn't. HIV, for example, there were people in this trial who had HIV. There were people who had congestive heart failure and CHF. So about 22, 23% of them had it. Well, let's go over the efficacy first. If you look at this, the vaccine group had 11 cases. And again, there were about 13,900, so just roll it up and say 14,000 in each group, each arm. So 11 cases, that's less than 0.1%, had COVID in the vaccine arm versus 185 or 1.3%. So if you wanted, you could look at it that way and do what would basically be 1.3, 100 divided by 1.3, so about 50, unless I really screwed that up for a number needed to treat. But take a look at the incidence, which is really what we're looking for here. 3.3 versus 56 per 1,000 people. And if you do that math, the VE or vaccine efficacy is 94%. Confidence interval says anywhere from a low of 89 to a high of 97. And I'll tell you, if I have to pick 89, I will get on board at that any day. So that's really pretty good. They also calculated it for severe COVID. The number of patients with severe COVID in the vaccine arm was 0, and thus 0%, with an incidence of 0. And then in the placebo arm, there were 30.
How long did you have to write the script for figuring that one out?
It was hard.
Okay.
There was a lot of R. Thank God I have a new computer to be able to process that. 30 patients with severe COVID in the placebo group for an incidence of 9.1, and the efficacy, because of that 0, was 100%. Well, so highly effective. One of the things that we did see is that the efficacy is lower in the folks who are older. And fortunately here, damn it, having the two of you here is really jacking me up.
We're so light.
You're not going to dive into the subtext there.
I know.
That was the whole last podcast that we did on Second Shift. So.
I know. Steve.
Yeah, tune in.
So let's go ahead and go to the next slide. Let's take a look at what the local reactions were and the systemic reactions. And keep in mind, if you're looking at these things and trying to determine whether it is worth taking the risk of these adverse events, I mean, I wouldn't like to have these things either. But remember what you're comparing them to, and you're comparing them to COVID. Yeah, the majority of people, not that one, Mikey, there should be one before it that has adverse events.
No.
Actually, you're right. My bad. Let's talk about this. Just back on efficacy. This is one of these things where if you look at, like Rimley said, you really, really, really don't need a whole lot of statistics. So let's take a look at this. What we're looking at is the cumulative incidence on the y-axis and time from your first shot on the x-axis. Blue is placebo, ain't got no protection, and the red is the vaccine. You can see they start to break off right before 20, and it is really, really clear. I'm a big fan.
It's not like subtle. You know, the Pfizer graph looked almost exactly the same. You know, and it's not like this is really tight, guys. This is really close.
I don't know if I can do this visually. I think I need to see some p-values. So take a look at the next slide now, Mikey. Mikey's like, no, you were mean to me. I'm not going to show you.
I got to tell you, it's a beautiful slide.
It is. I'm a big fan of the comic XKCD. So I'm going to show you their version of this. And he had, as he usually does, really insightful comments about vaccine trials and what you're looking for as in a clinical trial. So you really want to try to get data that's good enough that you don't need a statistician. Now, personally, I like having a statistician, at least a taco eating statistician. I like having my pal on there. That's really what we're looking at, folks. It's really pretty clear. All right, now let's take a look at the adverse events, Mikey.
Okay.
After I falsely accused you of pressing the wrong button.
I know. Jeez, these are, yeah, come on, man. And I was just going to say, I'm glad you're bringing this up because there's been quite a few comments. I've sort of gone back in here and people are like, what about the anaphylaxis? What about all the stuff? Tell us about that.
Absolutely. It didn't happen. There was no anaphylaxis. Let's take a look at what there was. So as far as I'm going to show you localized adverse reactions and systemic, these were the localized adverse reactions, and 58% of people had them. And again, this is with the Moderna vaccine. The onset of these was day 1, meaning the day that you got the vaccine. The typical or median duration was 2 days with the first dose, a little bit longer, 3 days with the second dose. The most common, kind of by far, the most common adverse event was injection site pain, soreness. It hurt where the stuff went in. 87% going up to 90% with the second dose. The next highest after that was lymphadenopathy. And if you read into the data, it's actually axillary lymphadenopathy on the side of the injection, which sort of makes sense. And then redness, swelling, or hardness. Those were the things that happened locally. Let's take a look at what happened systemically. And you'll see that also on this next slide, the majority of patients had some type of systemic effect also. So systemic effects, again, 58%. Nope, that's not it. 55%. There we go. Had a systemic reaction. 34% of those started on day 1, and then another 37 on day 2. The duration here, again, 2 days. And the most common was fatigue. 39% had fatigue with the first dose, 68 with the second. The other big ones were headache, 35% on dose 1, 63 on dose 2. Kendra, who gave me my shot, poor woman, was doing a great job running through all this, and I couldn't even feel the needle, but she had a headache, and that's a pretty common thing. So headache, you see some myalgias, arthralgias, nausea, and vomiting. All of them were almost all of them were more pronounced with the second dose than the first.
Was there any? They don't break it down as to sort of the time of onset. It looked like Liz Fagan was asking in the comments, average time to onset post vaccination of any of these sort of. So effects. I would imagine, like, if I'm a betting guy, I would say the injection pain is probably immediate. Yeah.
Well, so because most of the pain is not from the needle.
Right.
It's from the reaction as it's starting to percolate in the muscle cell. So all of the median duration, they only reported it overall for all of the adverse events, and that was day 1. So,
and I think it was based off the daily reporting. So I don't think they looked from hours. I think it was day by day.
Exactly.
But so what you're saying is that you're saying
the trial that a bunch of the stuff was on day 2. I want to say that I read that either in their paper or in the FDA piece that most of their symptoms were on day 2. But I'm not. They can't. There's something reminding me of that.
Well, let's take a look. So I did see. Go ahead and go to the next slide, Mikey. We'll talk about the dreaded Bell's palsy. I see Samantha saying, "Does it hurt as bad as the tetanus vaccine?" No. It does not. Definitely not so much. So I see Gabe asking about.
Mine was actually very close to when I got my tetanus updated, but just saying. So for some people it was.
So let's talk about Bell's palsy because I've been hearing a lot in the interwebs. My county put up my picture. They sent it out to everybody with me getting the shot, and we had some interesting comments that popped up. Bell's palsy was one. Someone asked if I even knew what Guillain-Barré was. Yes,
Guillain-Barré?
I do. So Bell's palsy. there were some cases of Bell's palsy here. There were 3 in the vaccine group and 1 in the placebo group. And you're like, well, that's like 3 times as many. So clearly the vaccine causes Bell's palsy. All right, well, let's put this in perspective. There were 3 cases out of 13,934 versus 1 case out of 13,183. The percentage has a whole bunch of zeros after the decimal point. We looked at this a couple of different ways, and this is where Rimley and I were breaking out paper and doing just some algebra here. And the key thing you need to know is what is the base incidence? And Veer was talking about this. So the base incidence, if you just look in the general population, it is 3 cases per 14,000. We had to knock that down a little bit to get even denominators. It's usually represented as 25,000 per or 25 cases per 100,000. But basically what we're seeing, we would expect in the general population to see 3 cases of Bell's palsy for every 14,000 people. In other words, it is at the expected rate. Now, if you want to look just at this without thinking about the base rate fallacy, that's okay. You can do that. And you can even calculate, I did a chi-square, despite Rimley saying she was going to hurt me because of it, but I had to because these numbers are so big. I think the p-value was 6, 0.6. So it's just not a significant difference. So there were all cases in the Pfizer, but it is base rate. Yeah.
I mean, I was going to go out and get saline injections to prevent Bell's palsy after I read this paper. Should I not do that?
Probably not. And the reason is because if you look at death in this trial, there were 6 people who died in the vaccine group and 7 who died in the saline. So your saline may protect you from Bell's palsy, but at the risk of dying. So probably not a good plan. All right. So with both of these trials, just as we're kind of wrapping up, there are some limitations, and the limitations are just functions of the studies. Number 1, we do not have data on pregnancy, and we do not have data on kids. We don't have great data on prior infection. We do want to talk about that a little bit. We are not collecting data because the only people that the case definition was symptomatic COVID. We can't really talk about asymptomatic positive test or asymptomatic transmission as much as we would like to. And then we also can't really talk about duration of immunity. Now, having said that, we're not going to let that stop us, and we're going to pontificate about it here as we talk about some of these myths. So I think that's bottom line. That's what the Moderna trial was. Someone, I think it was Samantha, was asking about, well, how do you compare the two? Which one would you get? I would get the one that's available.
Down the line, I would probably say, once we reach full availability, I don't see a huge difference between the two.
No.
So
yeah,
I was just going to say, so it's great. I think we've done a good job of covering the Pfizer. We've talked about Moderna, but is there anything else we can compare it to? Are there any other vaccines out there that we could say, well, what are normal issues with some other vaccines that are out there?
Well, I mean, I think
one over to Ritu.
Yeah,
lobbying motion.
I think one of the questions that one of the comments that I heard a lot from people is this was really rushed. They didn't do a complete study. Yeah. This was not the same level of scientific rigor. And so I was being a nerd. I decided, well, is it? I don't really know. So I asked some people. I'm like, and yes, number 1, is this really fast? Absolutely.
Warp speed.
Ain't no vaccine ever happened this fast. So that is an interesting question and an interesting concern, but there was no scientific shortcut that was done here. And so, I went back and the best way, I'll just throw, let's say we've got one vaccine that's got 40 that comes up before the FDA. It's got 40,000 people in the trial, was 95% effective, something like 80% of people had a reaction, but most of them were minor. Maybe 20% had a fever, no serious. We got another trial with 30,000 people in it, 95%, 94% effective, same percentages. And then I'm like, okay, well, let's look back. Well, here's another trial vaccine, 30,000 people in it, 90% effective. 22% of the participants had a fever in 24 hours. So almost twice as many as in these other two trials. And FDA approval, no problem. And that was the Shingrix vaccine or the shingles vaccine. So being a complete and total nerd, I actually went and read the freaking FDA briefing on the when GlaxoSmithKline got the Shingrix vaccine approved, and the numbers and the percentages were remarkably similar. 30,000 patients, less than 50, it was like 29,500 patients, 14,000 in each group. No long-term effects. Any serious adverse events happened in the first few days. There were no, and serious included like a fever. There were no anaphylaxis. Nobody died from a direct vaccine component. And then the other interesting thing I hear people say is, well, only a few hundred people in the whole study got COVID. So how do you make a decision based on only a few hundred people? Looking at this vaccine, it was 450 people across both groups that got shingles. So again, all of the parameters that you look at for this are very similar to previous big vaccine trials. And so I think that's really an important thing. Now, the one difference, because there was no like worldwide shingles pandemic and there was no multi-billion-dollar checks being written to study this, it took them almost 5 years to get 30,000 patients enrolled in this trial. And that happens because if it's going to cost you $1 billion, spread it out over a few years, that saves you some money. And then getting volunteers to sign up for a trial is one thing when you're like, well, I don't know, I've never had shingles, I don't care, versus, hey, help save the pandemic. So I do think that some people have voiced concern, well, they've only followed people out since July, although phase 1 was way back earlier in the Moderna trial. And Moderna has a lot of data on mRNA. They were getting ready to launch a MERS trial when this all kind of came down. But I think that one difference is that we don't have years of follow-up. That being said, when you read their methods, it's almost exactly the same. They checked in with people every day for all of 7 days, and then they relied on other reporting tools for other. So the methodologies that were followed in these trials are exactly the same as any other sort of modern vaccine. And I got a lot of, when I actually read the stuff on other vaccines, I felt better about it too. There were no shortcuts in what they went through to complete this, other than the original plan of these trials was to follow people out for a year. We haven't followed them out for a year. So one of the reasons why the FDA didn't give final approval, well, there are a couple of reasons. One, from a process perspective, it's a lot quicker to get an EUA than it is to get final approval. Two, they won't give you final approval until you've completed the study as per you set out to. So part of the study is to follow those people out for a year. And I think that, once they do that, the FDA will probably turn around and provide approval. The interesting thing too is, though, there's all this discussion about what to do with people who got the placebo, because that table that Jeff put up is so gorgeous. If I were a participant in the trial and I was in the placebo arm,
be like, what?
Now there's EUA. You don't know, but I would want the vaccine at this point. And so there's been a lot of discussion between both Moderna and Pfizer about how to get people who were in the placebo arm vaccinated. so that because of the ethics of keeping them unvaccinated when you now feel very strongly that there's a positive impact from vaccine. Yeah. So I feel pretty comfortable that there were no shortcuts here.
Hey, we got to get Dr. Crow out of here. She's got another engagement. She was nice enough to jump on and hang out with us for a bit, but we've flapped our gums for 1 hour, 32 minutes and 39 seconds so far. And she's got to bounce. Thanks for jumping on, Remley. Appreciate you so much.
Thank you all.
Remley, thank you so much. As always, it's wonderful having you with us.
Yeah.
Pleasure nerding out with you, Jeff.
Thanks, Remley.
We'll catch on to the next one.
Right. Have a great night, Remley. So now that we have Remley off and she's not there to correct my math, let me go ahead and get the number needed to treat. I did go back. I noticed he was asking for number needed to treat to prevent 1 hospitalization, 1 death. Unfortunately, we can't give you that because that's not the outcome that was measured. So I did calculate the number needed to treat for 1 symptomatic COVID infection, which was the primary answer here. And that's 83. So it's a difference between it's actually 100 divided by 1.2 minus, or 1.3 minus 0.1. So that came out to be 83. And if you look, so still a little bit more effective than, say, statins for primary prevention. I think it's worth it.
So Jeff, you know what I find really interesting about calculating the NNT is that it's really a snapshot, right? Like the NNT, if we get good vaccine penetrance, the NNT is going to go up quite a bit. And if we let things rampage for a few more months, then the NNT is going to be a lot shorter, a lot smaller, right?
Yeah.
And so I think it's important to keep that into consideration too, is that it really depends on that incidence of disease in the community, really.
Absolutely. I also calculated the NNT to prevent 1 severe case, which by definition is a hospitalization. So I guess you can do that. And that is 1 divided by 30, or 100 divided by 30. So 3 to prevent 1 severe case. And that's with the numbers as they are now. Before we hop on, Mike, move on a little bit, I do have to say that the best way to visualize this is if you create a waterfall plot chart using R in Texas, because Texas is a delegated practice state. You will clearly see that you should stop intubating hypoxic patients.
Yeah. For those of you that are playing along with the either Second Shift or EMS Lighthouse Project Podcast bingo card, you therefore should be either drinking or getting your, I think using the F word is the free spot, we decided. But yeah. Jeff mentioning that Texas is a delegated practice state is definitely on your card somewhere. So make sure you get that checked out.
What's funny is Texas being a delegated practice state is actually going to help our vaccine distribution quite a lot.
Oh, yeah. Yep.
Yeah, because you don't have to worry about a scope of practice.
Yeah.
We do worry about a scope of practice. It's just whatever we want it to be.
Right. Luckily, our scope of practice
for everybody
to do these injections during a pandemic. So
yeah.
So that was actually explicitly put in there.
Yeah.
And the chair of the scope of practice committee here in our state on this very podcast. So there you go.
Well, thank you very much. Thanks for that little shout out.
Yes.
Hey, Mikey, yeah. I saw another question in there that I don't think the folks on this podcast are really able to answer, but I think a great question is, can employers mandate this vaccine?
I wonder if there's another place to learn about that.
Yeah, I was just going to say, I will channel my inner Samantha Johnson and I will say, it depends. How about that? Well, here's the thing. You can, go listen to the Standard of Care podcast, episode 3. I believe it's called Ouchie. That hurt. That is the podcast. But they're also going to be live. Wait, watch this. You want to see something really cool? Watch this. Watch this. Oh, no, not that one. Is it this one?
You have to push this button and then that button?
Yeah, I got to push this button. And then I'll just do this. I'll take myself out for a second. But I wanted to share with you their little...
I want to see it.
Got something. Hold on. It's coming right here. Let me see if I can do this. No, that's not going to do it. See, there's too many buttons to push. That's the thing. Is like,
We need like an actual engineer.
All right, boys, let's go ahead and start tap dancing. We're going to cover here.
oh no, I'll get it.
Do
This, and I'll just do this. You see, and they were making fun of me because I always say this. I go like that, and then I hit this thing right here. And there's a really long delay tonight. I don't know what's going on. There it is. Are you guys with me still or no?
Well, we're waiting on something to happen. But
I see the
there we go.
post there. Yeah.
Yeah, there's a really strange delays because it was almost automatically up there for me, but for you guys didn't even react for like 20 seconds. But anyway, standard of care, they're going to be live right here wherever you're watching. They're going to be live here next Tuesday. And they're going to do an update to the podcast that they did about can your employer mandate you and all these different workers' comp things and all this kind of stuff. So tune in wherever you're watching right now. You can come Tuesday, 5:30 Pacific, 8:30 Eastern Standard Time. You see, I threw a little wrench in there because standard of care people are all Eastern Standard. So we got people all over the damn place right now.
Because the most important thing is to get the people on the podcast actually on the podcast. So.
Yeah, exactly. Exactly. So anyway, if you want to learn about the legal aspects, listen to that episode 3, then tune in right here on Tuesday, and we'll get this sucker going for you guys.
Well, so there were quite a few questions, including from my sister, who just had a delightfully adorable baby, about pregnancy, want to get pregnant, or I've been pregnant and I'm breastfeeding. Can we get this? Well, if you look at the science, meaning the clinical evidence, the answer has to be we don't know, because they exclude pregnant patients. Now, typically when it comes to things dealing with OB-GYN, I like to know what my limitations are, and that ain't the kind of doctor I am. Don't ask me about OB-GYN stuff, and for the love of God, don't ask me about rashes. So I go to ACOG. ACOG is the American College of Obstetrician and Gynecologist, and take a look at what they said. In the show notes, we're actually going to post a link to the ACOG recommendation, but they have a very lengthy discussion, and basically it says, look, no great evidence, but we don't see a reason not to. We can't recommend that you do it because there's not the evidence, but we really are not seeing a reason not to get vaccinated, whether you want to be pregnant, whether you are pregnant, or whether you're breastfeeding. So I think that's a really important one. We talked about the issue with kids earlier. I haven't seen anything from AAP about this. They have a tendency to be fairly conservative. So my guess is they're going to say we just don't have the data, which is a very truthful statement. And then before we hit on the myths, I think that'll be kind of fun. I do want to hit on something my director, Mike Knipstein, pointed out to me, and that is the CDC has issued some guidance on something I think we're all going to face. If we go back and we take a look at what those localized effects and systemic effects are, let's go through some of them. Fever, fatigue, headache. Those things might be confused with COVID. And up until now, as soon as you have those, we exclude you from work. So we have the potential to exclude, if we go out and vaccinate our entire workforce on a single day, and this is about to be why I would say don't do that, then you could lose up to half of your workforce. And now you have a mission problem. So number one, space out how your folks are getting vaccinated. Number two, the CDC recommends, or they put out some recommendations that says, hey, there are some really COVID-like symptoms that we don't expect to see with the vaccine: loss of sense of smell or taste, cough, shortness of breath. Those are not immune responses. If you have those, off you go. Do whatever your system has built up to deal with people who are suspected of having COVID, whether that is quarantine and test on day 5, or whatever your crisis staffing model says, do that. On the other hand, they're saying if you're having low-grade fever, if you're having fatigue, soreness, myalgias, those are very consistent with an immune response, which is what we expect. Give it a couple of days and then be reevaluated. So if you just have 2 days of these pukey kind of feelings from an immune response that are consistent with an immune response, you don't necessarily need to quarantine. And I think that's very useful advice for us.
Well, I would agree. And yeah, that's definitely going to be probably a hot topic here relatively soon. All right. Do you want to get into some of these myths real quick?
Let's do it. I do want to introduce, we covered this the last time. We have the Grinch, his buddy Max from
Max.
the Grinch.
Right, we know the name now.
It was Grinch. Yep, Max.
No, that's Max.
I thought it was Max.
Max.
Veer, you have the youngest. You're the one with the youngest kids.
It is Max with an X.
There we go. And the platypus, do you recognize that? Is that still a thing, Veer?
I recognize it. I don't know where it's from, unfortunately.
This is Perry the platypus from... Oh, gosh.
Oh, now he's stuck.
I am. The 2 kids and the mom and the...
I don't see it. All right, I was Perry the platypus.
Perry the platypus.
Somebody GTS it. Jesus, criminy. How hard is it?
Somebody will help us. It's a cartoon that's actually really good. With that in mind, let's hit some myths. What do you think?
Right. So a lot of these we've already covered, but is it true that it messes with your DNA and is going to cause Frankenkids?
I wanted to do that one just because I wanted to say Frankenkids. I just think that's such a cool concept. No, this is
Fire bad. Oh, sorry.
That's brains. Phineas and Ferb. There we go.
Ah, yes.
Oh.
Yeah.
No, but Frankenstein fire bad. That's a whole thing.
Yeah.
Yeah. Fire.
So I think we covered that.
Because they light him on fire in the original Frankenstein.
Now I got it.
Right.
All right. Yes. No. I think we hit Bell's palsy, Mike. What do you think?
We did hit Bell's palsy. And we did talk a little bit about anaphylaxis, but I mean, at least go ahead. I mean, we said that it, at least in the Moderna trial, there were no episodes of that, but that doesn't mean people, with Pfizer, we've seen some. I think there's been 2 in Alaska. So people are having anaphylaxis, right?
Well, so remember that was outside the trial. Maybe it was anaphylaxis, maybe it wasn't. We just don't know because we don't have great definitions. And I'm not saying it wasn't, because the truth is, whatever the side effect, no matter how rare, when you give it to 100 million people, you expect to see some cases of it.
Yeah, I think that's the summary.
I think that one of the things that you have to understand is, I think it's a losing battle to say, well, there was none of that in the trial, because some things could be so exceedingly rare that you don't see them until you vaccinate 100,000 or 1 million people.
Yeah.
You still have to acknowledge the fact. That being said, if the incidence of something is 1 in 150,000, the sort of relative risk at both the individual level and certainly at the population level is such that it still makes sense to continue.
And that level of risk exists with a lot of things that we do, right? And I think that's something to put into perspective. If you have 15, 30,000 people that it doesn't happen, and then you get a couple cases out of 1 million, that happens for a lot of stuff, and we recognize that. And it's not to say that it's great or that we want it to happen, but it is looking at that population level. And that's something I think we've been trying to do a lot during a pandemic.
Yeah. I think one of the things, the fact that something is happening with some of these people, right? Like we said, we don't have all the data. We can't be sure exactly what it was. But those are the kind of reports, the 1 in 5 million, that are going to keep some people from getting the vaccine, right? So what can we do or say to help those people understand, and realize that the benefits of getting the vaccines, like at least for now, seem to extraordinarily outweigh the risk of getting COVID?
I mean, I think that's it's a risk benefit analysis. I mean, everything I do in the emergency department has a risk benefit. I have patients who come in and demand TPA for their stroke, but then won't get the vaccine. I mean, the risk benefit in TPA for stroke is almost exactly the same. The number needed to treat, the number needed to harm are like the same number, but the number needed to harm in the vaccine is still going to be exceedingly small. And I do think, I think that if your response to concern for this is, oh, you're just being silly, it's fine, it is frankly doesn't work. Right. Because there is risk to everything you do. There's risk to getting the vaccine. There's risk to ordering your food on DoorDash, Veer.
Not being able to answer the door because you're on.
There is risk to crossing the street, and at both a population level and an individual level, you have to basically evaluate that for the group as a whole and for me as an individual. And are there going to be people that have reactions? I would be willing to bet there are. There are 330,000 dead people in this country from COVID-19, and to me, that risk benefit ratio is not even close. So, but you can't just there's to the point where there's funds set aside for people who have bad outcomes. Now, to that being said, the preliminary safety data on this stuff kind of blows everything I've ever read out of the water. It's amongst the safest vaccine I've ever seen or kind of reported.
Well, I think that's a great segue into the next question or the next myth or the next.
Can I interrupt one question before you do that, though?
Oh, yeah, go ahead.
Yeah.
Nick put a comment in there, and I just wanted to respond to that. Yes, there was a lot of screaming at my house during the last Mandalorian episode. A lot of screaming.
God, we just second shifted it. Anyway, what were you going to say? Something, Dr. Jarvis?
I was just going to compliment you on your segue ability.
Well, thank you.
I know, and I totally blew it out.
You did. You just jacked that up.
Jeez,
I hijacked that.
I'll give this one to Dr. Vithalani. So, along the terms of what Dr. Sahni was just talking about, right? We know at least maybe that short term, you're going to have pain at the injection site. There have been some reports of an anaphylactic type reaction, but we don't know anything about long-term effects. So, how do you sort of help ease that thought process, or that, because that's a real concern that people have got?
Yeah, I think so. And there's a couple things I like to keep in mind. So one, the phase 1 data has been continuing, right? So there were a number of people that got the vaccine very early on to look for that. And that data is still being collected. But two, there's some information out there that basically says, especially when you look at vaccine trials, I think all the effects happen within the first 6 weeks or so after the injection site. And I think that's something to keep in mind. There was 2 months of a requirement for post-second dose data. There was 14 weeks for some of it in the Pfizer trial, at least. And so I think that's part of it. But to Dr. Sahni's point, I think, there's always going to be risk associated with it. I think one of the best sort of posts that I saw about the pandemic in general was that, the general public are not used to watching the scientific process unfold. Right. And so you have to take all of this into consideration. And we're, as humans, we're used to looking at a very micro level of how does this affect me as an individual person. And you definitely want to look at that when you're making decisions for yourself. But there's also the larger picture of all the bad stuff we've gone through this year and what this really means from a moving forward perspective and what it really equates to when we talk about large scale risk. I think that's really how I sum it up in my head. That's why I felt comfortable going and getting my vaccine. And I think that, it is a calculated risk, especially as we talk about those that weren't in the study population and looking at people that might want to get vaccinated but weren't directly studied. It's really the same thing. It's a calculated risk, but we know what effect it has on people. So.
Yeah. Did you have anything to add to that, Dr. Jarvis, or anyone else?
No, I think you did a great job as usual.
Awesome. All right. Well, I'll throw this one to you then, Dr. Jarvis. And I'm using air quotes here. I've already had COVID. Why should I go get the vaccine?
So this is going to be an intellectually unsatisfying answer. I will tell you that. There are a couple of things that we can lump into this basket, and perhaps Drs. Vithalani or Sahni have a great answer to this. I don't. So I've had several medics that have had it. I had one who was hospitalized. And that's their big question is, do I need to get this? Well, my first answer is that if you look at the vaccines, or the trials, they tried to exclude people with prior history of COVID, so they can't directly address it. Next, you can look at how long does the immunity last from the shot. A big question is, do we need to get boosters like we do for flu? Couple of components to that, and I think we ought to hit that. And then the other thing is, how long does natural immunity, if you get it, how long are you immune from it? Well, let's address that first. Because we can't directly answer this question, we just have to make inferences. So let's kind of go over the data that we can use to infer. First off, we know that immunity from having COVID is not absolute by any means. We know that you may continue to test positive for around 90 days, even though you're not transmitting the virus. But we also know that that's not the same thing as 100% immunity. CDC sort of says, hey, you got a warranty for about 90 days, but that's about the extent of it. The main reason we know immunity is not 100% is because we're seeing reinfections. It's not a huge number of reinfections, but we are seeing them. And it's not just false positives. Here are the reasons we know they're reinfected. Number one, they test positive and they're sick. They're better and they test negative. Then they get sick again and test positive again. So that's one reason. The other reason we know is we actually did serotyping on these folks, and it's actually a different, there's been some genetic, and I don't know whether this is antigenic drift or shift. I could never remember those. I saw that on the MCAT and went, next question, C. So we're seeing some mutations. And if you look at the news coming out of London, where they're seeing huge increases in this strain, you can still get that. So we are seeing some differences, and that natural immunity doesn't last. We are seeing some signs, particularly in the activation of B cells and T cells, that immunity may be longer lasting. And because of that, the CDC recommends that even if you have had COVID, once you are recovered, you go ahead and get vaccinated. I don't think that's an ironclad. That's just best available evidence, and that's really all we have. There is, let's see, what else can we say about that? Let's talk a little bit about mutation, because I think that is a big concern. We are seeing, let's talk about flu. Why do we get a new flu vaccine every year? It's because it mutates, and it mutates in a way that decreases the effectiveness of the prior year's vaccine. And that's not even accounting for the fact that it's not just one flu virus. There are multiple flu viruses out there. So it changes enough that an immunity you had before isn't going to be good for that new strain. It's not that your immunity is waned, because if in the future that same strain comes back around, you still likely have protection. It's just this is a different strain. With the coronavirus, and remember, there are lots of coronaviruses out there. We've all had one before. It's the common cold. There is less mutation. Doesn't mean it's not mutating. There's just less of it. The key thing, though, and this is the way I like to think about this, is that the way the vaccine gets into our cells to work its little nefarious things on us is through the spike protein. That's its key to the lock. Well, our antibodies are triggered to the spike protein. So if it mutates so much that the spike protein now is not recognizable to the antibodies. It also ain't likely to be recognizable to the lock, and therefore it won't be dangerous for us anymore. So that's the thought. The bottom line, though, is we don't know. It's all inference on that. We don't think we're going to have to get a new COVID shot every year.
But I guess we'll find out.
Exactly.
Well, we'll have to find out. And I mean, I think that goes to the whole point, too, of, people are like, can I take off my mask? Can I do all that? And to me, what drives our community's response is what COVID looks like in our community. And so we can take off our masks when we no longer have COVID in our community, or check that. And when we no longer have community transmission of COVID, you may still have you still may have some COVID in your community that pops up, but as long as you can, if you get it under control and then can do good contact tracing, then you can actually remove your mask. We can go back to the way it was, and the whole point of getting the vaccine is to build towards that. But what I tell people who ask me about what to do after you get vaccinated is that the community's approach to COVID is based on how it looks in our community. And that getting vaccinated is one of the tools to reduce those numbers so that we are less likely to have community transmission of coronavirus. That's how I look at it.
I think that's perfect.
Yeah, I totally agree.
Well, good.
So I saw a comment in here about, is there any merit to getting an IgG titer to ensure that you're not being injected with the same strain to which you already have immunity? And the thing that I wanted to point out is, remember that this is different than other vaccines. Other vaccines, for example, polio, is you would have a live virus type of injection. That ain't what this is. You are not being injected with a virus in any way, shape, or form. There is no virus in that syringe. The only thing that is in there is a little bit of bacon grease surrounding a messenger RNA. And that then tells your body. Oh, Ritu, you're going to die here. What's your bacon grease comment, man?
You just made me think of bacon.
Ah, good.
And I just now I do popcorn in bacon grease. It's freaking delicious.
There you go.
Wow.
So, yeah, now I have, anyway, you're not being injected with a vaccine. It's the spike protein, and the spike protein is exactly the same on all of these strands of SARS-CoV-2. So the new thing bouncing around London, exact same spike protein.
Yeah, the IgG is not going to differentiate between the different strains of coronavirus. Yeah. It's a genetic analysis that does.
So another question Dr. Fagan was asking was about how long should you wait after you've had the active infection before you get immunized. And I think you probably should not get immunized while you're sick. How long? I don't think there is a great answer. CDC is leaning towards 90 days. Dr. Fagan works at the same hospital system I do. She's up in the Metroplex, Dallas-Fort Worth area. And she says they just put out something. This is why I should check my emails on Sundays. But that says wait 90 days after getting COVID. But she's also saying if you happen to have COVID and are not all that sick and get the bamlanivimab, we're calling it Bam Bam in around here. If you get the Bam Bam, the monoclonal antibody, how long do you have to wait? So 90 days. I think it's a good rule of thumb.
But to be clear, you don't have to wait 90 days after you've had COVID. They just say you don't have to get vaccinated for about 90 days. So they're saying as long as you're beyond the infectious period, you can get the vaccine. But the key is, if we have our little EMS agency of 4 of us on the screen, but we only get 3 vaccines, and one of us has had COVID in the last 90 days, give it to the other 3. I'll get, if I had COVID, I'll get mine a little bit later.
So that's how I.
Yeah, they were very clear in their webcast that Mike and I both watched, that they said that you can get the coronavirus vaccine. Their only recommendation is to not get it if you're still in this sort of infectious period. Was their only recommendation.
Well, thank you for pointing that out. Even as you were saying that, Liz popped up and was correcting me also. So thank you for pointing that out. And I think we just don't have great evidence on this particular thing. What was the other question that was? Nope, gone. Oh, I do know they are seeing some people. Yeah, no, I don't know where I was going with that. Never mind.
Wow. I think on that note, we're 2 hours and 3 minutes in. Can we call it a night?
So I do have to wonder whether we beat our airway episode or our epinephrine episode. What do you think, Mikey?
God, they're all close. I'd have to double check, but they're all about 2 hours. It's important stuff. All right. We've managed to keep 60 or 80 or 100 people on here, and they're interested.
I like it.
Yeah.
So
right.
Bottom line, we tease this up by saying, find the facts that you need to decide whether you're going to get it. Obviously, the 3 of us decided to get it, that it was worth it. Mikey, hopefully you will have an opportunity shortly.
Yeah, for sure.
And then hopefully y'all got the information you needed.
Yeah, exactly. All right. So with that, I will get us out of here. On behalf of Dr. Jeff Jarvis, Dr. Ritu Sahni, Dr. Veer Vithalani, you've been watching the EMS Lighthouse Project podcast. We are a proud member of the Flybridge Ed Podcast Network and a Fire Dog production. We will see you on our next live, which is, by the way, this Tuesday, Standard of Care. Check it out. All right. We will see you guys on the next one. Have a great night. We'll see you.
Good night.